Try to wrap up the morning, and I'll try not to keep everybody from lunch for too long. So I wanted to spend a short period here and talk about some things beyond sequencing, and that I think are actually important in terms of understanding communities, particularly complex communities, but as well as simple communities. So the community that I'm going to talk about is actually a very simple community, and it's a quite interesting community, as you'll see. And it's a community that's associated with whiskey, and that's something that I like. That's something that I know several of you like as well. Now, whiskey, of course, is one of a number of spirits that are aged after they're distilled. The material after it comes out of the still is put in a barrel. It's usually an oak barrel, and these are stacked in warehouses, and they're aged anywhere from 3 years to 20 years, and at that point they're taken out and bottled. Now, during that process, alcohol in those barrels is lost to the environment through evaporation, and that's a necessary part of the maturation process. Now, it turns out, and this has been known for a long period of time, that neighborhoods that surround these areas, over time, start to develop black discolorations on exterior surfaces. So this is a talk about the built environment, but it's a talk about the built environment outside of the occupied space. So this is the exterior of an aging warehouse in Lawrenceburg, Illinois. Here's a blue door on another aging warehouse in Ontario, and a fire hydrant, and you can see on the chain link fence and so on. This is, we use kilometers per hour in Canada, and here's a sign that's just about obscured by this black fungus. And the black fungus even grows on stainless steel. This is an old fermentation tank that's like in the back lot of one of these warehouses. I had some photographs of boats and other things that were stored back here that are also covered with the thing. But more importantly, and why people have recently become concerned about this, is it also grows on houses in neighborhoods that surround these things, and in some cases for a radius of maybe 1 to 2 miles around these ethanol emitters. So my introduction to this work came maybe about a decade ago, and since we've done quite a bit, but the introduction was an aging complex that consists of 14 warehouses in southern Ontario, and it's I guess maybe about 8 miles or 10 miles east of Detroit. And they age Canadian whiskey. Some of it's actually very good Canadian whiskey. From the air, if you look at it, it kind of looks like a printed circuit board where the things that would be the computer chips are the aging warehouses. So you can see each one of those, and they're divided into cells that contain different amounts of barrels. And each one of these warehouses is surrounded by a moat that can be flooded. Of course, this is very expensive stuff, and if a fire were to catch, they don't want the entire thing to burn up. So we were particularly interested in looking at these areas, and as well the surrounding areas. So there's just the location there and its proximity to Detroit. Now, one challenge in trying to understand the spread of affected area of this phenomenon, we'll call it, is that people tend to clean their houses. And if you have a darkly colored house, you don't maybe clean it as often if it becomes affected by this as a light colored house. But it's difficult driving through a neighborhood to really understand what the dispersion is. So others, and us as well, have looked at material surfaces that are independent of cleaning. Particularly, we've selected the scaffold branches of light colored trees, silver maples and Norway maples, and these are quite commonly planted as urban trees. And if you examine those scaffold branches and just visually look at them and assess them against a rating scale in terms of 7 or lower, maybe about 6.5 is the normal kind of gray color of these branches, with 10 being completely black, and you can actually see that there's a fairly consistent pattern that matches with what one would expect of the ethanol dispersion profile of the warehouses. And in fact, if you look at some of the dispersion models, that's consistent. And you can just use your nose because the odor threshold of alcohol is around 1 part per million. And as you walk through these neighborhoods, you get the welcoming smell of whiskey. So when you take a sample from one of these surfaces and scrape it and sprinkle it on a Petri plate for years and years, interested folks have done this. You get a long list of things that one wouldn't really necessarily expect, like alternarian or Basidium and Cladosporium and Penicillium and Aspergillus and things like that. But when you look at the actual physical manifestation of this growth, it just doesn't really seem like it's any of those things. If you look at it using molecular methods, this is using cloned 18S product, and then you match that against GenBank, you come up with a list of all sorts of things that are variously plausible, not completely. Now, if you scrape the material and look at it directly under the microscope, what you find is that the vast majority of it looks about all the same. These roughened, thick-walled black cells. And there was a fungus described from Cognac, France in the 1880s that was illustrated. It looks like the same thing. So our first step in trying to understand this community was to look at it in a microscope and see really what it consisted of, what was growing there, not necessarily just what had passively deposited there. So as I said, if you sprinkle it on a Petri plate, you get a whole bunch of colonies of all kinds of things. And you can, if you're familiar with these things, you can probably spot the alternarias and the Basidiums and the Cladosporia on here. But if I zoom up on this little square that I've got highlighted, these tiny little colonies with the arrow appear to be the thing that's really predominating and growing on this material. And if you zoom up on that even further, after it's been plated out and allowed to germinate, you can see that cell, that group of cells. It actually looks morphologically quite consistent with what we were seeing just in the original scrapings. And you see it germinate here. You can pick it off with a pin, and it grows very slowly, but you can grow it up. When you grow those things up for weeks, they look like this. They're tiny little nondescript black colonies. If you look at them microscopically, they sort of produce spores or broken ankle fragments, but they don't really match to anything except that old illustration that had a name that was in the genus Torula, which was simply something that was reserved for anything that no one really knew what it was, and it was black and rough, and that's that. It hasn't really been brought up to date much more than that. So when you take those cultures and grind them up and do some Sanger sequencing of some meaningful loci, this is nuclear small subunit ribosomal DNA, and build that into a phylogeny with things that are closely related, you can see plating out at the top is this thing that we've described in the genus Bauduinia, with the type as Bauduinia complutensis, which is based on the old name from 1881. Its closest relative is appearing to be around the genus Prepaniomyces, which is another one of these colonists, black colonists, but this one from rocks in Antarctica. It probably occurs elsewhere, but no one's really bothered to look very closely at these kinds of communities. Anyway, these are in the Capnoviales, and the family is probably the Stratospheriaceae. So since we began this work, we've actually looked at a number of geographic localities and collected specimens from which we've been able to derive cultures with patience and growth media amended with alcohol and some other tricks. And when we've taken those cultures and done some further investigation using multi-locus phylogenetic approach, this is just one gene. This is EF1 alpha, partially F1 alpha, and you can see we actually have multiple lineages of this that in fact correspond quite closely to geographic distribution. So the one at the top is the type species from France. Orientalis we found from the Far East and Korea. Caledoniensis is from Scotland and elsewhere in the UK. Panamericana we found all the way from Canada through the US, Central and South America, and then Antilleensis is from the Caribbean. And those are quite consistent, and we can isolate those fairly reliably, and certainly can find them by looking directly at genes. Now, the question is, what is the fungus doing there? One of the advantages, I should say, to having a culture in hand is that then you can do experimental work with it. You can understand how it responds to controlled environmental changes, and from that point, hypothesize a little bit more about what it might be doing. That iterates back into the ability to look at RNA libraries and things like that. So one thing that we found is that this fungus, it does occur on surfaces that are subject to episodic wetting, episodic exposure to ethanol, and high temperatures that are periodic high temperatures, usually diurnally. So it has an inducible mechanism of heat resistance. So if it's preconditioned at a sublethal heat exposure, then say 37 degrees, then if you transfer it to 52 degrees, it's happy to grow. But if you just transfer it directly to 52, it kills it. This inducible mechanism of heat resistance can also be similarly induced by a brief exposure to ethanol vapor. So that seems to be one mechanism that this fungus uses. There are others, including ethanol as a germination initiator, and to some extent ethanol as a carbon source. But with these organisms in hand physically, then one begins to be able to do this kind of work to understand their relationship with habitat a bit better. Now, we have now, fortunately, in the last year, a full genome sequence from the, it will actually be the American species of the fungus, and that's allowed us to look even further to understand some of the mating type genes and things like that. So I'm going to step back to the community as a whole for a moment. Now, you can see from the slide of the Petri dish that I showed that a number of things occur in this community. There are lots of fungi that live there, or at least are visiting there, and then there's one thing that seems to predominate. And when we did some denaturing gradient gel electrophoresis, you can see in the lane on your left-hand side, a dark band here that probably corresponds to our Boulder group of things, and then a few bands. So it seems to be a relatively simple community. And indeed, when we look at it using 454 pyrosequencing and ITS2, you can see that this is just 3 samples out of a couple of dozen that we've run, sorted according to reads just of these 3 samples. You can see that the signals from members of the genus Boulder by far are the most common representatives that we're finding in the community. So in absence of the ability to amend, in this case, the UNITE database with well-validated sequences of these organisms, those would be unknown. So really, in terms of getting a handle on that community, being able to establish microscopically what's there, and then being able to recover that, characterize it, and iterate it back into that database to classify the community is very helpful. So as I've started to work on this, I've developed search patterns for things that are of interest, and I've started to look at buildings a little bit more closely from the outside. I have a lot of history with buildings on the inside, but from the outside. This is the building. I was actually an undergraduate in this building. This is the old botany department at the University of Toronto, and you can see there's quite a bit of black accumulation on the bottom of this. I've looked at it. Some of it's algal. There's some fungal material there as well. But it's quite interesting because there's copper flashing on the building, and you can see along the copper drip lines that it's clean. So I have a colleague in geography who's interested in deposition of carbon-based air pollutants on surfaces, and she's assured me that all of these things are air pollution. I said, no, air pollution doesn't respond to copper like that. That's microbial. This is the building where I am. In fact, my window is around the corner from this one, and you can see even on the entrance to my building, we've got a similar kind of phenomenon going on. So I don't have to go too far from home to see these kinds of things. And if Paula's in the audience, here's one that's kind of close to her. It's the Waldorf Astoria in Manhattan. So the couple of take-home messages that I wanted to leave you with is that new fungi, and this is probably generalizable to microbes, occur in all sorts of places where we least expect them, and that many habitats that we typically consider to be well characterized may actually not be so. There's an awful lot of fungal biodiversity that's historically been ignored because it doesn't have morphological features that allow it to be differentiated, and that's really concealed in these quite simple organisms. You really need a combination of culture-based and culture-independent methods to get at some of these things, particularly where you have organisms that are important but not well characterized. And in the case of ethanol here, it seems like VOCs may actually have some important function as habitat determinants in fungi. So that's all I have to say. And we can lead into lunch now. I'd like to thank the organizers for a fantastic meeting so far, and a bunch of colleagues who contributed to this work. Thanks very much. questions?
Yeah, I want to, I'll address it loud enough so everybody else can hear. And no, I can do it directly. But I thought it was a terrific presentation. I just want to make a point that there is a community that's concerned with damage to cultural artifacts associated with air pollution, mostly acid deposition and things of that sort. But it seems like your work would fit really well into helping
I've been
address that.
connected with those guys and have reviewed their grants, and I've dug into some of their sequence libraries. And the fascinating thing is that I've not been able to find representatives of these things in their sequence libraries.
Yeah.
So it's you know, it suggests to me that the things that are growing on marble monuments in Milan and Rome probably aren't the same kinds of things that are blackening these kinds of environments. It seems like this is a little more distinctive. But yeah, it's interesting. There's definitely a lot of work on that.
Right.
So it's interesting that, you know, when you went back to it, it was like the majority. And then when you figured it out by, you know, I don't know what any of these things are doing. So it strikes me that, you know, like Greg was talking about the open reference OTU picking, that what you're having is all these sequences getting dumped into an unknown apparently. And what would be interesting, and this is other people could use this, is to say, I'm going to use this, you know, I have my ones I know and the ones I don't. Let us cluster. I bet if you went back and clustered those again, with his technique, you'd find this OTU cluster that was massive. And that would be a way to say, hey, I've got this guy here who's always on these samples, and I'm going to go look for it, right? Because it's clearly, see, am I missing something, or was it really fun?
Even as Greg gave that talk, I was taking notes as fast as I could type, because it really, you know, it really struck me that is an answer to a number of these problems we face, especially in these communities that are not very well known.
Well, it's a general problem, though. I would point out for everybody's study, if you've got this issue where you have a sample and you have this thing that's really fun, you know nothing about it, and it's an indication you should go and look for it in the environment through fish, through microscopy, through culturing, through scraping, and find out what the heck it's doing and advance our knowledge of a particular environment, because that's just brilliant. That's just
it's really been fun.
Yeah.
Can you get Carrie in that?
Yes.
Yell, Carrie.
Ethanol production facilities is another place to look when they dry the mash. You not only have ethanol, you have acetic acid and acetaldehyde. We actually ran biofilters as an air pollution control system, but that's another place. We're all over the United States now, and that'd be another place to go look.
Interesting. Yeah, we've got it from bakeries. We've got it from several commercial bakeries as well, but they're a little bit better ethanol controlled than the emissions on these warehouses. Anything else?
There was a paper at ASM looking at ethanol, acetone, and two other chemicals in outdoor air and the impact on some microbes. So
Interesting.
I can find it.
Okay.
I blogged about it actually.
Okay, thanks.
microBEnet.
And now it's time for lunch. So we go